Hello guys and welcome back to Excel Share Academy. So we were doing about the microtubules and we saw like how alpha, beta tubulin monomers come together to form a heterodimer and to form the microtubules, right? So let me just summarize. So we have also talked about that it is a hollow tubule having the 25 nanometer in diameter, and there are 13 protofilaments which together come and forms your microtubules. Okay. Now, in animal cells, most microtubules extend outward from the centrosome, which is located adjacent to the nucleus in the interface, right? Now, during mitosis, these microtubules similarly extend outward from the duplicated centrosomes to form the mitotic spindle, which is responsible for the separation and distribution of chromosomes to the daughter cells. So we know that in the dividing cell, we have got the centrosomes present here like this. Okay, so first during the division, first centrosomes gets duplicated. Okay, and then your from here, based on the gamma tubulin, at that base, okay, using gamma tubulin ka base, yours microtubules are extending outwards like this. Okay, and यहाँ पे आपके chromosomes रहेंगे to which it attach. Okay, so now like in the previous lecture we saw that when there is a catastrophic the depolymerization is happening, okay, at the plus end, then that's how these motor proteins are carrying your chromosomes in the respective directions. Okay, so this is the same which just now I have made. So centrosomes are there, and from there microtubules are extending, and the plus end of your microtubule will go and bind to the chromosomes. Okay. Now, the centrosome serves as the initiation site for the assembly of microtubules in animal cells, which then grow outward towards the periphery of the cell with their minus ends anchored in the centrosomes. Now, this can be clearly visualized in the cells that have been treated with the colcimate to disassemble microtubules. So, if you remember colcimate, we have discussed in the previous lecture that it's a drug along with colchicine, which helps or which arrest the cell at that particular stage. That is, it does not allow them to divide. Okay, so that's why they are also used as the anti-cancer agents. Now here, if you see in this figure, this is a normal case where the microtubules are distributed throughout. So these lines, these are your microtubules. Now, whereas in this case, the cell is treated with the colcimate. So here you don't see any extension. Like after 1 hour with the treatment of the drug, it just extended only this much. Can you see the difference? यहाँ पूरे cell में फैला हुआ है आपका microtubules, right? But here it is not. And then later the drug was removed. Okay, so what we saw is that from the centrosome only to a few length only the microtubules were able to assemble or grow. Okay, so that's how your colcimate will inhibit the polymerization and hence inhibiting the cell from growing. So microtubules grow by the addition of tubulin to their plus end, which extend outward from the centrosome towards the cell periphery. So the role of the centrosome is to initiate the microtubule growth. So yes, so where you have got your centrosomes present, from there itself the microtubules will start. तो ये प्लस एंड हो गया जहां पे और ट्यूबुलेंस ऐड होंगे. ठीक है. So the key protein in the centrosome is the gamma tubulin, so which is present at the base. Okay, which is helping in the nucleating the assembly of microtubules. अब actin में कौन सा था? It was your formin dimer. Okay, now analogous to this, in the case of microtubules, it is your gamma tubulin. Now, gamma tubulin is associated with 8 or more other proteins in a ring-shaped structure called gamma tubulin ring complex, which act as a seed for rapid microtubule growth. So if you see, there are certain ऐसे capping proteins onto which your gamma tubulin is present. इस पे आपका ऐसे alpha beta tubulin molecules comes like this. ऐसे 13 आएंगे and 13 rings ऐसे protofilaments form होंगे. ठीक है? So that's how your microtubules are formed. Okay. Then are you able to follow? Any further doubts are there?
No, ma'am.
Okay. So the centrosomes of most animal cells contain pair of centrioles. oriented perpendicular to each other and surrounded by pericentriole material. So what they're saying is that inside the cell, you have got these centrioles. So they're present like this, perpendicular to each other. And from here only your microtubules are originating. Okay? Now they are present inside the pericentriole material. ऐसा रहता है. This whole together, the centrioles plus the pericentriole material together is known as the centrosome. So centrioles are cylindrical structures containing 9 triplets of the sorry, they contain 9 triplets of the microtubules organized around a central cartwheel-like structure. So if you take the centrioles ka cross section, so here 9 triplets of microtubules. So it means like this. ऐसे 9 it will be present. So 9 is 27 microtubules will be present over there. Okay, so that is like one centriole. So another centriole will be also like that. So they also function at the cell surface as a basic bodies of cilia and flagella. So again, itna yaad rakhna that microvilli, villi structures, everything, they have got the actin filaments. Okay, now the cilia of the bacteria, flagella of the bacteria, they all are made up of microtubules. Okay, however, while the centrioles are necessary to form cilia and flagella, they are not needed for microtubule organizing functions of the centrosomes and are not found in many unicellular eukaryotes. Okay, so what we are saying is here that apart from cilia and flagella, okay, centrioles are present in the centrosomes, but every eukaryotic organism does not have centrosomes. Okay, for example, the mouse eggs. Now, it is a pericentriole material that contains the gamma tubulin complex and function to initiate microtubule assembly to anchor the microtubule minus end. Okay, so it is the pericentriole material and not directly the centrioles also. So यहाँ पे आपका centriole present है, then in the pericentriole material you have got the gamma tubulin जो आपका base बनाएगा. Okay. Now, if you ask what is the then real function of centrioles, still now that research is still going on. We don't know. We just know that in the centrosome, you have got the centrioles, and pericentriole material. In the pericentriole material, you have got the gamma tubulin, जहां से आपका microtubule शुरू होता है. ठीक है? Okay, so this is the electron micrograph of the centrosomes, which is showing here the microtubules. Yeah, see, जो ऐसे line से दिख रहे हैं, those are your microtubules, which is all originating from your centrosome. Now here, this is centriole, which you know always present like this. Sorry, ऐसे, ऐसे, ठीक है? So अगर ये आपका ऊपर की तरफ खड़ा है, then it is ऐसे. Okay, now this area is your pericentriole material which is surrounding your centrioles. Now again, this is a transverse section of your centriole. So this I was telling that there are 9, 1, 2, 3, 4, 5, 6, 7, 8 and 9 triplets of microtubules are present. Okay, so this is how it is present and they're arranged. Now coming to the maps and the organization of microtubules. So what are maps? It is microtubule associated proteins. Okay. Now these microtubule associated proteins are going to help in stabilizing the microtubule plus and minus end. Like they ensure that whether the microtubule should keep on extending or it should get stabilized to that particular length. Okay. Now maps control dynamic instability by binding to microtubule plus end, but others act to sever the microtubules or stabilize the microtubules by binding along their length. So we have got maps. Okay, ये आपका microtubule हो गया plus and minus end. So they are helping in stabilizing your plus end. Okay, there are many other proteins which bind over here in order to stabilize a protein. So microtubule stability is also regulated by extensive post-translation modification of the tubulin. And what kind of post-translation? It can be phosphorylation, acetylation, palmitylation, or removal of addition of C-terminal tyrosines or glutamines or glycines. Okay, so ये थोड़ा सा याद रखना. So what we are saying is maps are helping in stabilizing the microtubule. Okay, now this stabilization can be regulated by post-translation modification. What kind? Phosphorylation, acetylation, palmitylation, or removal of C-terminal tyrosine or glutamine glycine. ठीक है, तो ये तरह के post translation modification है, which is going to help in stabilizing your microtubule formation. Now, there are different types of maps present depending on the cells. For example, map 1, map 2, then tau protein. So we know about the tau proteins, right? So tau proteins is like, we say that if they get misfolded, okay, they result in the neurological disorder, for example, in the case of the Alzheimer patients. Okay, so tau protein, which is present in neuronal cells, is actually a type of microtubule associated proteins. Okay, so we have got in the neuronal cells MAP1, MAP2 and tau proteins, and MAP4 is present in the non-neuronal cell types. Sorry. So the tau protein has been extensively studied because it is main component of one of the characteristic lesions found in the brains of the Alzheimer patients. Okay. Now, who can tell me like what exactly happens in the Alzheimer patients? Like we know that it is a neurological disorder, but what exactly is happening? Anyone? Who? Okay, Akshay has joined. Akshay, do you want to comment?
uh,
Hmm.
uh, there is protein misfolding in
Okay.
the brain,
Yes.
so which blocks the neurons. So the neurons ultimately start degrading. So that particular part of the brain where the memory is stored, like that part of brain gets degraded a bit. So that's why the Alzheimer's patient don't remember anything.
Yes, yes. So when we say the proteins is getting misfolded, that is your tau protein only getting misfolded. Okay. And it is responsible to carry out the impulses. We'll see like how it is helping this how this protein is helping in carrying out the functions. Okay. And this is like once it gets affected, it spreads to the nearby neuronal cells. And because of which the connection is gone. And as she said, so finally, the cells are lost. When those neuronal cells are lost, obviously the memory is lost. Okay. Yeah. A good example of the association of stable microtubules with cell polarity is provided by the nerve cells. Now we know that in the case of the nerve cells, axons and dendrites are present, right? So both axons and dendrites are supported by stable microtubules. Guys, this is really important. Okay. So the microtubules in nerve cells are not anchored in the centrosomes. Okay? They are not originating from the as centrosomes. Okay, they are just released from the centrosomes and then they are aligned in the dendrites and axons. And they are stabilized by the capping proteins. Okay, so I'll show in the figure like how they are stabilized. Okay, so here you can see this is your neuron, right? And then we have taken the axon body and the dendrites are present. Now we see that the each end of your microtubule, so you can see that it is not originating from the centrosomes, right? It is released from the centrosome. Now they are not anchored to the centrosome. They are just released from the centrosome and they are stabilized. It means what? They are not growing, they are not depolymerizing. Okay, so these microtubules neither polymerizing, neither depolymerizing. Why? Because they are stabilized by the capping proteins which is present at both the ends, plus and minus, plus and minus. Okay, and they are associated with the MAP proteins. MAP, microtubule associated protein. So in the case of the neurons, we have got the MAP2 which is present on the dendrite. Now, if you see in the structure here, what do you see in this case and in this case? What is the main difference apart from the association of MAP2 and tau protein? What else do you see the difference?
Ma'am, polarity.
Very good. Yes. So here we see that it is plus minus, that both are in opposite direction. Correct? Okay. And they're associated with the MAP2 in the case of the dendrites. Now, whereas in the case of the axon length, again capping proteins are there. The polarity, if you see, both are in the same direction. Okay, so plus here, minus here, plus here, minus here. Now here also you have to understand that the minus end is towards the nucleus, or we can say that the plus end is away from the center of the cell. ठीक है, ये चीज है जो आपको याद रखना है. Did you get it? So map 2 is associated with your dendritic microtubules which are present in the opposite direction. Okay, whereas in the axon it's your tau protein which is associated with the microtubules. Now if anyone asks like what is tau protein, so tau protein is nothing but your map, microtubule associated proteins. Okay, so they are present in this way in the axon region. Okay, now going ahead, this is the same thing which just now we have discussed. Let's just go through. So microtubules are organized by distinct maps in axons and dendrites. And in axons, the microtubules are all oriented with their plus ends away from the cell body. Yes, just now we checked, right? Now in the case of the dendrites, the microtubules are oriented in both directions. Some plus end points towards the cell body and some are pointing towards this periphery. Now, these distinct microtubule arrangements are paralleled by differences in the maps. For example, axons में आपका tau proteins है, but there is no MAP2. Whereas dendrites contain MAP2 and there is no tau proteins. Okay. Now, because of these differences in the map distribution, both they play an important role in stabilizing the microtubules in the axons and the dendrites, and hence they have got the differences. Like dendrites का क्या काम है और axons का क्या काम है. Okay, now we'll see about the tau proteins. So as I said, tau proteins kya hai? It is part of your microtubule associated proteins. Okay, they play important role in maintaining the stability of the microtubules in axons in the neurons of the CNS. Okay, now this is how it is arranged. Can you see what is alpha beta? Alpha beta, alpha beta, alpha beta is your tubulin, right? Tubulin. So ये आपका microtubule हो गया. This is your microtubule. 1 second. Okay. So this is your microtubule. Now what is kinesin? Kinesin is your motor protein, correct? And here your tau protein is present. So the arrangement of tau protein is that along the entire microtubule, okay, the tau proteins are arranged. This is how, see, can you see? This is alpha beta tubulins are there and it is helping. Now, if you ask what is the function of tau protein, it is to stabilize the microtubule. So आप देख सकते हो यहाँ पे उसने कैसे अच्छे से बांध रखा है through all the tubulin which is present, right? See, what you have to understand is like why the stabilization is needed, because in those axons and dendritic regions, we don't want microtubules to be depolymerize or polymerize. जो एक बार fix है तो fix है, ठीक है? It is not like the other cells जो you know ऐसे rapidly बार ऐसे change होते रहते हैं. So neuronal cells, you know that if they change means our memory changes, everything changes. So here these tau proteins are present which is helping in stabilizing the entire microtubule. Okay, and you can see on the top of that the kinesin is bringing the vesicle or whatever the cargo it is having. Okay, so there is this video, just look, just watch this. You'll get an idea like how the tau proteins are present and how when there is a misfolding happening, how it is affecting the neuronal cells and ultimately resulting in the Alzheimer's. Let me know if you can hear.
The healthy brain.
Are you able to hear?
Yes, ma'am.
The healthy brain has around 86 billion interconnected neurons which communicate using electrical and chemical signals. Tau protein, through binding at its repeat domain, supports microtubules needed for cell structure and to provide rapid transit pathways for vesicles, mitochondria, and cell components to the synaptic junction. Aging brain neurons lose some ability to clear waste material, which can accumulate as lipofuscin, where tau can bind and misfold. Misfolded tau can capture normal tau, causing it to misfold in the same way and form oligomers, which disrupt the synapse. Tau oligomers can transfer to other neurons and initiate the same process of tau capture, causing the disease to spread. As oligomers aggregate and grow, they form paired helical filaments and then bundles called tangles. As neurons fill up with tangles, they can no longer function properly and eventually burst, leaving behind just tangles.
Okay. Did you get it? Yeah. So we saw like how the tau proteins are arranged and all the vesicles, are being transported with the help of the kinesin. So what is happening? Why those two junctions were shown? See, when we say that the impulse is transmitting from one neuron to another, it is the electrical or the chemical signals, correct? Chemical signals matlab kya hua? It is the release of the neurotransmitters. Neurotransmitter kaise release hoga? Aapka vesicles se, correct? So if you remember, we always said that the neurotransmitters are present in the vesicle. These vesicles comes here and fuses with the presynaptic neuron, and with the help of the exocytosis, the acetylcholine is released outside, right? And then it will go and bind to its receptor and the signal is carried out. Now, as you saw, the microtubules were present over here, and on the top of these vesicles, the kinesin is bringing all this. Okay, so if you remember, kinesin, I said that it moves towards the plus end, and now we just saw that in the axon body, the microtubules are arranged plus in this direction, minus in this direction. Now in the video you saw like many kinesin proteins were walking, right? They were transporting the vesicles in this direction. Now what had happened? Because of certain chemicals, the tau proteins were misfolded, right? And when this misfolded was there, all those kinesin were not able to move properly. It got blocked, and the neurotransmission was blocked, and these misfolded tau proteins were also transferred to the another neuron, and eventually the entire cell then finally dies. Okay, as I said, the tau protein eventually will form the tangles, and when they become the tangles, it means what? It is like high level of misfolding which the chaperones cannot refold, and that's how the cell dies and only tangles are present, and then it spreads to the entire neuronal network, and that the entire region gets misfolded, and hence those particular region now cannot be regained. And that's why the people with Alzheimer they can never remember those regions if the neurons are lost. Okay, did you get it, guys?
Yes, ma'am.
Yeah. Okay, so there are many medicines which people are trying, scientists are trying to cure Alzheimer. So what in the case of Alzheimer, like high level, when they say that this is the last stage in Alzheimer, also you have got the different stages, like starting and middle and the last stage. Last stage is gone where the where people don't know how to eat, how to breathe, even they forget that, even though we know that breathing is involuntary, right? But it is then when it is not in your control, then people forget how to eat also. That is the last stage, and it is very difficult to deal with such a adult and not knowing to do the stuffs like routine stuff also. So in the early stage, if it is detected, like you keep on forgetting, like आदत रहती है ना हम लोगों की भी बोलने की कि अरे ये भूल गए उम्र हो रही है, that we are forgetting stuffs and all. So that's how you need to understand the correct signs of this, like whether you're really having this disease or it was just a mistake. So one factor is that you can go is with the age. Okay, why with the age? Because the chaperones, all these proteins are not present in high concentration in the old age people. Okay, and there are less chances that these tau proteins will be getting refolded back. So that's why we say that with the age we tend to forget stuffs. Then one small assignment, or if you know the answer, you can tell me the difference between the dementia and the Alzheimer disease. Right now, anyone who knows the answer? Shruti, Tanvi? Yes. Okay. So both are different, guys. Okay. Huh.
As far as I know, Alzheimer's is a prion disease, while dementia is more about memory. I'm not sure about the proteins involved for dementia. I don't know whether they are.
Okay, just try to get the answer because in both the cases it is the memory loss. Okay, in both the cases it is memory loss. Both are related to the old age only. But yeah, dementia
but
can happen to people at the age of 30 also. But this is like rare cases. Yes. Yes,
okay,
Shruti,
okay.
you want to add something?
No, no, no.
Okay. Just go about it and search. Okay. See, when we study something, we should know all this thing. Abhi, dementia or Alzheimer, many people thought it is the same thing. It is not the same thing. It is different. Alzheimer is worst. Dementia is still, can be curated or at least can be controlled. Whereas Alzheimer cannot. Okay. Microtubules, motors, and the movement. Microtubules are responsible for a variety of cell movements, including the intracellular transport. So this already we have mentioned that they are involved in the vesicles and the organelles transport, right? And also the beating of cilia and flagella. There are two members in this motor proteins, kinesins and dyneins. Finally, we are talking about kinesins and dyneins, which I was discussing from the beginning of this topic, right? So let's talk about these proteins. Okay. Let me show you this video.
Kinesin is a motor protein that moves vesicles or organelles toward the plus ends of microtubules. Kinesin consists of two heavy chains wound around each other in a coiled coil structure and two light chains. The globular head domains of the heavy chains bind microtubules and are the motor domains of the molecule. Kinesin is
Okay. Okay, so that was just the introduction to the kinesin. It was like present in the text in the reference book. So that's why I added. So now you know that it has got the heavy chains and the light chains and they are coiled together. So this is the head domains, which is actually responsible in the walking onto the microtubules, right? With the help of the ATP. So with each step, ATP is being used. Okay. Now, the light chains are responsible for binding to the vesicles. Similarly, here you have got the dyneins. So when you see that in the lengthwise, dynein are small and kinesins are bigger. Now, dyneins walk towards the minus end, kinesin towards the plus end. Please remember, okay, this is important. So here they are showing minus end की तरफ जा रहा है आपका dynein. Again, ATP is required. These two are the heavy chains, and at the top you have got the light chains to which the vesicle or the organelles will come and bind. Okay. This is the same like how vesicles carry the how kinesins are carrying the vesicles and moving towards the plus end, and if it is a dynein, then it is moving towards the minus end. Okay. Let us watch this video also. Are you able to see the screen? The movement of vesicles along microtubules in vitro. That's the title of the video.
Yes, ma'am.
Okay. There is no sound in this. Can you see this thing? Slowly, slowly it is moving. So it started from here and now it is moving like this. Here also. This is moving. This is moving. Finally, it has gone. Okay. So these videos are the ones, they're the original ones, like, with the help of the electron microscope in the live cell, they have actually captured this all videos. Okay, that's all guys. Any doubts for today? I'll just stop